Wow, what a conference. Really incredible. And I have a difficult thing to do, which is on the afternoon of the second day when everyone is finding their glucose load kicking in from lunch, wake everybody up and get them to think about therapeutics. We have a therapeutic opportunity, and usually when I speak to angel groups, that's a little difficult for them to comprehend. But I think this group might be the exception because of the strong healthcare base and all of the physicians in the audience. What I'd like to do is suggest that we have an opportunity that is later stage than most things that you'll see, because we've been able to raise so much non-dilutive capital and be so capital efficient, and where you can ride on diligence already done by institutional investors. In other words, what we've tried to do is, because we're in a very technical space, we've tried to make sure that there are other people in the deal that you can look to and say, well, that's a professional investor in this space. They've got the MD, PhD kind of people that can evaluate that and hopefully take some comfort from that. And then finally, we are in JLab, so we do already have a strategic follower. We do have a $16 million award from the state of Texas, but I need to tell you we've spent half of it. So about half of it is left for 2018, and then the award will be over. A safe harbor statement, just to let you know, we'll be talking about future things that I can't guarantee. Well, what are we? We're an oncology-focused biotech company. We drug what has been a target of interest since 1982, the Wnt beta-catenin pathway. This is a fundamental embryonic stem cell pathway that we use in our bodies very actively when we're kids, and as adults it's mostly shut off. But cancers reawaken it, and it is a primary mechanism for cancers to mutate and resist the therapies we throw at them. It doesn't matter if it's immune oncology therapy, if it's a targeted therapy, if it's chemotherapy, it's all the same. Now, the drug has broad potential across many solid tumors and heme tumors, but this is typically in late-stage disease. So it's a combination therapy play, and while that's tremendous for commercial potential, it's not great for capital efficiency, because what we want is orphan approvals where we can use the FDA's accelerated approval path and get to market in 3 years instead of 8, because we're a biotech and we have to raise the money that we use. So we have actually found 2 of these where the drug looks very exciting as a single agent in oncology, and that is a very high hurdle to find. Now, we actually have filed our IND. We're in discussions with the FDA right now. I actually had to respond to questions yesterday during the meeting, but the questions look very innocuous, and we're very pleased, and we expect to get our IND approval probably the end of next week or the week after. Clinical trials probably start 1st of February or so. The broad potential of the drug is that this pathway is turned on and heavily activated across all of these tumors: colon, breast, lung, melanoma. This is the reason that checkpoint inhibitors stop working in melanoma. It is one of the key pathways that is turned on in virtually across all the hematologic tumors when they're in blast crisis. And so this is really a very broad therapeutic. In addition, we have 2 areas that we're very excited about for registration. And our registration strategy is to get the drug to market as quickly as we can, because once you're marketed, then as you do additional clinical trials, patients are paying for the drug. You've got an income line. You're not working entirely on other people's money. It's a very good place to be. So the first area is desmoid tumors. And desmoids is an unusual orphan tumor. You can think of it as scarring that has become cancerous, and it is 100% due to one oncogenic driver, this one, beta-catenin, which we named the company for, the bottom protein in the Wnt pathway that drives this tumor. And interestingly, very rare in oncology, this disease cannot mutate. This disease is 100% driven by this. Now, we've done work with what models are available in this disease. And here I'm showing you some cell strains where our drug is 10 times more potent than any other drug and kills cells 4 times as fast. And also the only animal model, which isn't an exact animal model for the disease, but it's the best we have, where the drug has very strong performance as well. The second area is a very difficult disease called osteosarcoma. This is obviously a bone cancer. And our drug does not work that well in the primary tumor. We'll get tumor regression, we'll slow it down, but we won't cure it. What's interesting, though, is because this is a bone tumor, the primary tumor can be cut out with surgery. That's not the death-defying event. The death-defying event is 2 to 5 years later when you come back to your sarcoma expert with lung metastases, and there's nothing he can do for that. And this is where it turns out this is a very Wnt-driven disease, and this is where we are seeing incredible efficacy in our preclinical models. So here I'm showing you in mice, curing mice that have these lung metastases in virtually 80% of the time. We take the mice lungs out and look at them, and we have virtually no lung mets at all left in the mice, but quite a few in the control group. And interestingly enough, what we're doing right now is it turns out that large dogs, companion animals, get this disease, and although it's more aggressive than the human counterpart, it's quite similar. So we're actually doing right now a clinical trial with veterinarians in companion animals. So this is a natural tumor. It's not any sort of, super created artificial animal models. Actually, dogs being treated by veterinarians. And we have some just very beginning data, which is that we have one Great Dane where we have seen in the primary tumor an objective response. Now, I don't know that I would expect this response to continue because, again, our activity is in the mets more than in the primary tumor. But if you look at this series of radiographs, you can see in the last one, dated 29th of November, and the 46% just has to do with the magnification, that the tumor appears noticeably smaller on the right, and you can see the bone more through the tumor, suggesting that it's thinner. So we're very excited to see what comes out of our companion animal trial. We've also done mouse models of metastases, and this is a triple-negative breast cancer, where we basically infused breast metastases into the mouse and then allowed them to grow in the lung and look at what's happening with them. And you can see at day 38, the metastases are starting to take off, but the Tegavivint, our drug, treated arm, we don't see anything going on, and you can still at day 45 see quite a bit of suppression, as in the vehicle you start to see the lung mets really taking off. Now, I have to have a competition slide. Now, it's a little difficult for me for this reason. This pathway was discovered in 1982. It was given a Nobel Prize because it's such a fundamental developmental growth control pathway. All of our competition has died along the way. And the reason for that is that there were no good druggable sites in this pathway to try and go at it with the technology that we had. So what people did is they either took poor sites inside the cell where they couldn't really get a drug to stick very well, or they went outside the cell with antibodies and antibody-drug conjugates. The problem with going outside the cell is this is a fundamental development pathway. And so when you go up the pathway at all, you start to impact normal stem cells and important biological functions. So the toxicity of doing that killed off all these programs, and my last competitor was AngelMD, and they shut down their program in March of last year. Now, John, why would you have the chutzpah to think you could actually do this? Well, the reason is new biology in the pathway was discovered in 2008. and we were able to take advantage of that. All these other programs started in early 2000s or late 1990s. In 2008, the first really druggable site that actually had a pocket that you could stick a drug in and make a difference appeared, and that was that the pathway was elucidated to show a second checkpoint in the pathway, this TBL1 or transducin beta-like protein 1, which actually does have that deep druggable pocket and is important at the very bottom of the pathway, close to transcription where you want to be. And our drug, Tegavivint, uses that new biology to drug the pathway. Now, every company has to have a team. This is our team. We have very deep and capable folks. How do we afford this team? Well, we afford this team by using a shared service model. Most of these folks, except me, I'm 100% 24/7 dedicated to BetaCat. Most of these folks typically are in BetaCat about 50% of the time, and that allows us to have a more cost-efficient model and yet have broad, deep expertise that we need. All these people typically 20 to 30 years in the business. I myself have been 40 years in pharmaceuticals, and all these people have deep expertise that I invite you to read and look at their resumes. Our business plan is really to get to the top of the leaderboard with the FDA, because the FDA today has a lot in terms of determining what it's going to cost you to get to goal. And if you are hitting unmet medical needs that are important, then they put you at the top. And that's being an orphan drug, getting breakthrough status and accelerated approval. And if you get there, then you can do a phase 1, and you can sit down with the FDA on the basis of that phase 1 and talk to them about an accelerated approval phase 2 that will allow you to get a new drug application or permission for marketing approval. And that's huge compared to go through the usual 3 phases of development, phase 1, phase 2, and phase 3, in terms of time, 8 years, and capital intensity, hundreds of millions of dollars. So that's very, very important. Secondly, we've worked very hard to bring in as much non-dilutive money as possible. We've brought in about $17 million total, but by far the largest has been from the state of Texas, which we have $8 million to apply to next year. And then getting investors where we have both VC investors and high net worth investors. I'm pleased to say that Sante Ventures led our Series A. We also had participation by Jack Gill's Goose Society and participation by the Houston Angel Network, all of which we deeply appreciated. And in our Series B, which we're raising now, we have identified 2 VCs that are interested in leading the round. We're discussing that with them next week at JP Morgan. We expect to get that nailed down and have the security ready to go. And we're looking for angels to join that syndicate at the same terms as the institutional investors to take us through clinical studies and get us to the end game. We have very robust IP and a long runway, and I can talk about that in more detailed sessions. Here is what the Series B funds. We basically have filed our IND, as I said. We are now trying to do our clinical phase 1 and phase 2 studies. Because we're in oncology, we go immediately to patients, and we can go to therapeutic doses relatively quickly. We have potentially 2 exit opportunities that we're excited about. And it's very hard for me at this point today to tell you what makes the most sense, but we will be looking at both of them very hard and coming back to our investor base in early 2019, at the end of 2018, to ask them what they have enthusiasm for. The 2 possibilities are, with a good phase 1 data set, we could exit in early 2019. And that'd be very common. There are a number of deals associated with that. Of course, they have a wide error bar. And the most important thing in getting a great deal in this business is a desperate pharmaceutical company. It's very hard to know who will be in that state of mind and really want to do a deal. So that's one exit point. The other exit point is to go ahead and take a major blue chip VC round of 25 million or so to do that phase 2 registration study, go another 2 years, and then sell the company when we submit the NDA to the FDA for marketing approval. There is no pharmaceutical company, and I say that as someone who was in one at a high level for 28 years, there's no pharmaceutical company that doesn't understand you will actually have sales 6 months from now. And that would be a bigger exit. So in terms of what we're doing, this is our entire financial history. It's available on the website, our seed round, our Series A, our Series B. Our Series B is set to a $15.2 million pre-money. That may sound high, but I would point you to the fact that this is an unusually late opportunity to bring to Angels. And actually, we've benchmarked that very carefully. We've made sure that it's a good discount to what comparable companies are at, and we've absolutely had no pushback from our institutional investors. Add to that 6 to 7.5 million from CPRIT. That gives us about 12 million, which takes us into the second quarter of 2019, and delivers completed data in our first trial and a good piece of the data in our second trial, plus some additional sort of proof of relevancy data. We're going to do some work in blood cancer and a little work in complex solid tumors so we can prove to the pharma company that our desmoid work, our osteosarcoma work, translates into the broad potential that the drug has. And we'll be able to do that. We will only have at that point in time about 10 million invested in the company. I can tell you that when I was at Lilly, it was nothing for Lilly to frequently have up to 40 million in at that point in time. Now, what would possible returns be? Of course, this is somewhat speculative, but we could exit potentially after the phase 1 trials, or we could exit after the phase 2 registration trial. Those two exits, obviously, the phase 1 exit is sooner. If we look at a 50,000 investment, what we would benchmark based on the industry standards is a return of somewhere around $700,000. If we went and did the additional work, raised another 25 million, we think we'd get a $30 million pre-money at that 25. It would be 2 more years, but that 50,000 might get more of a $1.75 billion, million return, or 35x thing. So that is the benefit of therapeutics. We cannot promise that they will always work. They tend to be higher risk, but also higher return. This is one of those high risk, high return opportunities. It does have an orphan avenue forward, but then you're not just set looking at orphan returns. You're set looking at the broader commercial returns that are possible. Either way, we will exit. I actually have run sales and marketing organizations. I can tell you I have no enthusiasm for doing that from a biotech company. We want to use the high leverage point that we've got in this part of the value chain and exit and let a big pharmaceutical company use all of their infrastructure to do what they do well, which is the later stages of the event. Thank you. Any questions?
Thank you, John. We're going to, if anybody's got a question for John, just because we're running a little bit long, if you don't mind doing them individually, just so we can keep things moving. But hang on, John, don't go anywhere. This is an important piece for you.
Okay. Oh.
We didn't deviate with this one. It does seem like pharma, everybody will say to me, says, well, you know, that's not going to fit with the early exit strategy. But this is an early exit strategy as far as we're concerned, with a great high return probability that balances the risk. And we really appreciate your presentation, and I'd like to present you with our award of the Catalyst Fund.
Charlie, thank you so much. And the support is deeply appreciated.
Good luck. Great.